form the alveoli wall that will be involved in gas exchange. They have thinnest cell shape, so the shortest diffusion distance across that cell. So squamous cell, probably around 300 million of those in each lung, and if you take out the lung and dissect out, I don't know how they figured this out, but I guess they measured the size of a pneumocyte. And then they said if you take them and you put them together and lie them out flat, you get 70 square meters, which is pretty huge. So that's the surface area that a normal lung has for gas exchange. And this is necessary so that you get enough oxygen into the blood to maintain the O2 demand during very strenuous exercise. If you lose some of those alveoli, then you get less oxygen into your blood, and you're limited in what energy you can then produce for heavy exercise. The distance that oxygen and CO2 have to diffuse across is about half a micrometer, so it's really small. Not all of that is the simple squamous cell. You've got the simple squamous cell, and you've got an endothelial cell of a capillary, and they share a basement membrane, so that reduces the diffusion distance. Plus, you've got a thin layer of surfactant, which we'll talk about in Sherwood. That is to help to decrease surface tension so that alveoli stay open even when they're under high pressure, like exhaling really hard. So there'll be other numbers in other books, but it's between Martini and Sherwood, they agree, so we'll just stick with 0.5 micrometers for this semester, but I've seen other numbers. Type 2 cells, if you don't know what AKA means, it means also known as. I had a student ask me that one semester, I guess people don't know. So also known as, just a short version of that, septal cells. What that means is they're in the septa, they're in the partition between alveoli, but they kind of stay out of the way of the capillaries, which will then be involved in gas exchange. So type 1s will be by the capillaries for gas exchange, and type 2s will be in the partition, but somewhere else where they won't be interfering with gas exchange. They're very important because they secrete the surfactant. The surfactant will then decrease surface tension of water. If you were in my 103 lab, you know that the surface tension of water is such that you can actually float things on the surface of water. Try this sometime. Get some tweezers, get a paper clip, get a clean glass and water, and then take your paper clip and very gently put it on the surface of the water. A metal paper clip will float on the surface of the water. As long as it's pure water, the water molecules attract each other so strongly that you can float a metal paper clip, which has quite a bit of density on it, on the surface. You've probably seen on the mainland there are a lot of bugs that walk on water. They use the surface tension of water to do that. Well, we can't have that surface tension in our lungs because the attraction of water molecules for each other are such that they will attract so strongly that the elasticity of your alveoli will just follow the water and they'll just close down. You can't gas exchange. You've got to have something in between the water molecules to push them apart. That's surfactant. So what is it? And I live this word, so I put it on the slide. Dipalmitidylphosphatidylcholine. Just break it down. You got dipalmitidylphosphatidylcholine. got other things in it. This is a lot of fat. So the palmitoyl, phosphatidylcholine are fats, and cholesterol is a fat. Other phospholipids. Phospholipids will associate with water, so will the choline part of this molecule. Glycoprotein, that's supposed to, I thought I changed this, but I guess not. So this is, the T is supposed to be in front of the E, so protein and sialic acids. That's what the composition of the surfactant is. So you've got proteins and phosphates and amino acids that will associate with water, and then you've got cholesterol and fat and the lipid part of the phospholipids that will associate with other fats, and they get and repel water, and so they get in between water molecules and keep them from attracting each other strongly enough to close the alveoli down. If you lose surfactant, people who drowned but survived because they pumped all the water out of their lungs, they still have respiratory problems because though drowning, the water has gotten in there and washed away some of the surfactant. So they've got to have some positive pressure, and they've got to have a machine help them breathe until their type 2 pneumocytes can replace the surfactant that's lost. Some people have diseases where they can't produce surfactant anymore. Some babies born prematurely before the lung is able to secrete the surfactant, they have respiratory problems and need to have assistance to breathe because their alveoli tend to want to collapse because they don't have surfactant, which means water surface tension. Every time the baby or the person exhales, alveoli collapse, and they will not open up with the amount of effort that can be applied by the respiratory muscles. The term for alveolar collapse is atelectasis. So kind of a fancy word. Means the alveoli have closed down and no longer able to provide the surface area for gas exchange. So alveoli will be made up of all these type 1 pneumocytes, very thin. They'll be associated with the capillaries in the walls or partitions. These are the septa. You'll have the type 2 pneumocyte. It's kind of a cuboidal shaped cell. It's secreting a lot of fat, so that's why they colored it yellow, because fat is kind of yellowish. They'll be out of the way of the capillaries in the type 1 pneumocyte gas exchange area. They'll secrete surfactant that will cover the surface of the alveoli. The surfactant also helps to keep the alveoli kind of moist because it attaches to some of the water, but it also helps to prevent water from getting into the alveoli at too great an amount. So it helps to keep the alveoli dry. If fluids collect in the alveoli, then you have a greater diffusion distance for your gases to go across. So you want to keep the alveoli relatively dry. Moist, yes, but not full of fluid. So surfactant does that. So remember, we have this net filtration pressure in our capillaries that wants to push fluid out into the interstitial space, which then would get into your alveoli. So the surfactant helps to prevent that fluid loss. We also have capillaries that are called continuous capillaries. Most of what gets across to the tissues is through transcytosis, meaning you have the endocytose on the side of the capillary lumen and then exocytose on the tissue side of the capillary. So these are not very leaky capillaries, which also helps to keep alveoli dry. Alveolar macrophages are cleaning out alveoli. They phagocytose old molecules of surfactant so the surfactant doesn't build up down there and clog the alveoli. And then any particulate matter that you suck in that gets all the way past your respiratory mucus and cilia, then the macrophages will phagocytose that as well. You get any pathogen down there, then they'll mount an immune response to the pathogen. The arteries are carrying deoxygenated blood, which is full of carbon dioxide from your tissues. And then at the alveoli, that CO2 will diffuse out of the blood into the alveoli. And O2, which you have just inhaled, will then go into the blood. There will be a concentration gradient for CO2, which looks pretty small in numbers. It's about 6 millimeters of mercury pressure, which is kind of small, versus a much higher pressure for the O2 gas to diffuse. But remember, CO2 is more soluble, so it equals out, well, it evens out, and actually CO2 has advantage in getting across the alveolar sacs. The pulmonary veins will collect the oxygenated and carbon monoxide, no, whatever, the decreased carbon dioxide blood into the veins and then carry it back to the heart to then go out to the tissue. So O2 will be in high concentration and CO2 in low concentration to leave the heart. So here's the diffusion barrier. We've got red blood cells, plasma, but the actual diffusion barrier for CO2 that is made up by the lungs is the alveolar epithelium, which is our type 1 pneumocyte, our capillary endothelium, which turns out to be another simple squamous cell, so again very thin, and then the shared basement membrane. So it comes out to be about half a micrometer. Remember that there is that layer of surfactant too, which is increasing that slightly above 0.5 micrometers. But that is a barrier, so if I ask you a question about the diffusion barrier, what makes up the diffusion barrier, you better remember surfactant, otherwise your answer is wrong. Okay? So surfactant is part of the diffusion barrier. Alveolar macrophages, I like that picture. They go anywhere they want to. They've actually tagged them in the lungs and found that they enter the peritoneal cavity. So they just work their way through the diaphragm, go into the peritoneal cavity. I don't know where else they go, but they can pretty much go wherever they want to. While they're in the lung, they're called an alveolar macrophage. When they get into the peritoneum, they will lose that name. So that's and their function might actually change depending on what tissue they go into. So pleural cavities and pleural membranes. The surface of the lung has a visceral pleura on it called, it says, sheet of simple squamous cells, a serous membrane. That is actually going to fold back and cover the lining and line the inside of the thoracic cage and the top of the diaphragm. The space between the visceral serous membrane and the lining of the chest wall has fluid in it, and that space space is called the pleural cavity. It's not an empty space. It has fluid in it. It also is a fluid that's pretty high in water content. So here water is an advantage. We don't want it decreased in surface tension. We want its surface tension there so that the outer layer of the lung sticks to the inside of the chest wall. And when your respiratory muscles pull the ribs out and your diaphragm descends, it pulls the lung with it. And then that will create a bigger volume in the lung. And we're going to learn an equation, if you haven't already learned it, it's called Boyle's Law, that you have your chest wall relaxed and your diaphragm relaxed and your glottis open to the environment. You're going to have a pressure in the lungs that Boyle calls pressure 1, so P sub 1. And he measures the volume and says that's volume sub 1. And then he changes the volume. So you expand your ribs and descend, the diaphragm descend, which then gives you a second volume. So that's V2 on the other side of your equal sign. You don't know what the pressure is, but all you have to do is divide P1 times V1 by V2, and that will give you pressure number 2 or P2. So Boyle's Law and some other physics guys are going to mess with your little minds on this next exam because you're going to have to manipulate some equations to figure things out. So visceral pleura, parietal pleura lines the wall. They're stuck together by high water content secretions that the serous membranes themselves secrete, and that puts this fluid into the pleural cavity. The pleural cavity is just that space, very small space between the two pleura with that fluid in it. The pleural fluid has got enough other things besides water that it's very slippery, but not enough of those other things to interfere with the water's attraction for water molecules. The purpose of the fluid is to keep the pleural membrane stuck together so they work together during inspiration and expiration, and also to reduce frictional forces. As the chest goes up and the diaphragm goes down, the lungs are going to be rubbing against those moving surfaces. And if the fluid was not reducing frictional forces, then the outer surface of the lung would get inflamed, and then you'd have connective tissue developing, scar tissue developing, and your lung function would decrease. Pleurisy is when that fluid does not reduce the frictional forces between the chest wall and the surface of the lungs, and you do get inflammation in that on those surfaces. So the pleural fluid is either the pleural fluid is in excess, but it's also abnormal, and so there's an inflammatory response, and the chemicals of the inflammatory response irritate nerves in the area and cause pain, and also the fact that the surfaces are sticking together and not sliding across each other will also increase pain. When you breathe in, when you breathe out, both, you'll have this painful sensation when you're trying to breathe. So here are the lungs. They're on either side of the heart. They're the pleural cavity, and since this is dead meat, there's no fluid in it. They've sectioned it, and so fluid's leaked out. But the fluid would be in that space. Would be the pleural cavity. If you look at the lining of the chest wall, it will have kind of a sheen to it, a very light, silvery kind of sheen. And you could just take a piece of tissue or a Kim wipe and you could just wipe it off the surface of the chest wall. You could do the same for the lung. You just take something that's kind of abrasive like a Kim wipe and you just pull the simple squamous cells right off. So it's pretty delicate tissue. All these openings that look like somebody went in there and poked holes in the lungs, those are your airways that are bringing air out to the reddish parts where you have your alveoli and a lot of capillaries. So what breathes for you during quiet breathing? That's what you're doing right now. Your diaphragm is probably in the guys doing the majority of the breathing, and girls, it's your external intercostals doing the majority of the breathing. Unless girls are trained to breathe with their diaphragm, they tend to breathe with their ribs, and guys tend to breathe with their diaphragm. One possible reason for that is that females tend to get pregnant. And when you get pregnant and towards the end of the third trimester, you've got 7 to 8 pounds of baby shoving all your viscera up into your diaphragm, it's not going to descend anywhere to help you to breathe. And it would be kind of hard to learn to breathe with your external intercostals at that point. And suddenly, oops, my diaphragm's not working for me, and I'm really dry. Mothers tend to get short of breath anyway because of all that. But anyway, that's one proposed reason why females tend to breathe more with the chest muscles than not with the diaphragm. The diaphragm actually makes a bigger excursion. I mean, it goes further in its movement than the chest wall does. But the result is that you get whichever way you breathe or with a combination of both. During quiet breathing, you use those muscles to expand the thoracic volume, and then you turn them off, and the muscles relax, and that causes your exhalation. So you've got a very efficient method of quiet breathing. You just have to work to get the air in, and then turn the whole circuit off, and the chest and the diaphragm will go back to the resting position, create enough pressure on the lungs to get the volume up. And that usually is about 500 ml of air in and out per breath. If you need to force air in and out during exercise, you're going to start to use neck muscles that are attached to the ribs, but usually are involved in stabilization of the scapula. Muscles, so you've got your serratus anterior, pectoralis minor, sternocleidomastoid, those things like that can help you inhale. And you got your abdominal muscles and muscles inside your thoracic cage that can help you exhale. And we'll be talking about those muscles again, and if you didn't learn them last semester, then you can learn them for this test, because I am going to be testing you on those muscles again. To elevate the ribs and the sternum, we've got muscles that will pull on the ribs to get the rib that is below closer to the rib that is above it, and that will tend to expand the ribs laterally, and then the sternum moves out a little bit, and then the diaphragm will descend into the abdominal cavity. They equate that movement to raising a bucket handle, that you get an increase in volume in that space that once was lying down against the outside of the bucket, and now you're increasing the volume by that additional space. I don't particularly care for that analogy, but it kind of gives you the idea that things are expanding and lifting out to the front. So I boxed in the muscles. I think I changed the boxes just slightly because I think I did it really fast before and I kind of cleaned it up for the presentation, but it really hasn't changed in what are included. We've got our purple box, which is diaphragm and external intercostals, that are providing the majority of your quiet breathing. None of the other muscles, although some of your textbook comments are implying that other muscles are involved in quiet breathing, there's a lot of disagreement about that. It's primarily external intercostals, diaphragm, and we'll just keep that as the muscles of quiet breathing. Nobody is trying to refute the fact that they are participating, but there have been people who've studied the other muscles that are included under accessories like sternocleidomastoid, and they say that those are involved in quiet breathing. But because pathologically when they're paralyzed, you get abnormal quiet breathing. I don't think that really qualifies as a good research proposal. Pathologically, they may be stabilizing, so when they're paralyzed, then things go wrong. It doesn't mean that they're contributing to the tidal volume. Okay, so let's stick with diaphragm and external intercostals for quiet breathing. They work to inhale, and you turn them off, and then you get exhalation. So it's you only work to inhale, and everything else is passive. Turn off the inspiratory muscles. The muscles go back to their resting length. The thoracic cavity goes back to its resting volume. That creates a smaller volume, which increases pressure, again by Boyle's law. And the airway, as long as it's open, the air comes out of your lungs. Accessory muscles are involved when you need to get more air in or more air out. So accessory for inspiration, anything that is attached to the ribs or your clavicle and your manubrium and sternum that can expand the size of the thoracic cage. So sternocleidomastoid, your scalenes, which are attached to your upper ribs, pectoralis minor, which is attached to your upper ribs, and serratus anterior, which is attached to your lower ribs. All of those, if pectoralis minor and serratus anterior, they're stabilizers for your scapula. So as long as your scapula is stabilized by your rhomboids and your trapezius, then you can use your pectoralis minor and serratus anterior to pull on the ribs. Normally, the ribs are being stationary, and the other attachment is your insertion. So what you're doing is reversing your origin and insertion. We won't talk about force couples again, but they are acting as force couples. So they're reversing the origin and insertion, and they're actually pulling on the ribs to expand the thoracic volume. Pectoralis minor will pull up, serratus anterior will pull laterally. And then scalenes, which are attached to the upper ribs, and sternocleidomastoid, which attaches to your manubrium and your clavicle, they will help to expand up a little bit in the vertical direction. For forced inspiration. Forced expiration is going to involve the muscles on this side. Internal intercostals, when they contract, they pull the ribs, the upper rib down towards the lower rib, which decreases the thoracic volume. Your transversus thoracis will pull on the ribs and pull them closer together. And external oblique, and actually internal oblique and transversus abdominis, they all contract at the same time, compress the abdominal contents, push that up into the diaphragm, which then shortens the height of the thoracic cage. So rectus abdominis, external, internal oblique, and transversus abdominis, contract them all at the same time, and they will compress the abdominal contents. That will push them up against the diaphragm and help with exhalation. Yeah.
I know that you said the external intercostal muscle and diaphragm is for inhalation. So for exhalation.
You don't use any work. All you do is turn those muscles off.
Oh.
And then the volume will go back to resting level, and that's your exhalation. That's for tidal breathing, 500 mLs in and out,
So
tidal volume.
the accessories when go that we saw like on the back.
For your expiratory reserve, yeah. So we'll be talking about those. If you're not in lab, those terms don't mean anything to you right now. But if you are in lab, wake up, remember those terms, because you're going to be tested on them in lecture as well. So to inspire more air, sternocleidomastoid and scalenes pull up, so does pectoralis minor, and serratus pulls out laterally and towards the back. The external intercostals are contracting for the first 500 mLs. They're not resting during forced inspiration. They're also contracting, but you're using these accessory muscles to get the extra air in. So external intercostals and diaphragm, they're on during inhalation. They are off to produce exhalation. So no work is done during exhalation except passive work. And then for expiration, forced expiration, you add in all your abdominal muscles, not just rectus abdominis, but external, internal, obliques, and transversus abdominis. And then you get your internal intercostals, which pull the upper rib towards the lower rib and compress the thoracic cage. And then transversus thoracis, that's inside the ribs, that will pull the upper ribs towards the lower rib. And along with that internal intercostal, decrease the thoracic volume. So some terms for breathing: normal breathing, normal quiet breathing is called eupnea. And we can then force breathing, and that is called hyperpnea. So eupnea, you're using your diaphragm, your external intercostals. 500 mLs is going in and out during this quiet breathing. Hyperpnea, you're calling in the accessory muscles, and you're increasing above 500 for inhalation, and you're also going to force out more than that extra and tidal volume. You're going to go beyond that, so you're going to go into your expiratory reserve volume as well for hyperpnea. Apnea, and the A prefix means cessation, nothing's happening. So apnea is no breathing, cessation of respiration. Some people have that when they're sleeping. Some people have it because of medications they're on. So there are times when people just stop breathing. If you hyperventilate and exhale a lot of CO2 very rapidly, that will stop your breathing. That's what free divers use when they're going down to chase a fish, is exhale a lot of CO2, and then there's no stimulus for them to want to breathe for about a couple minutes while they're chasing the fish, spear the fish, and then they can get back up to the surface and go back to breathing again. So apnea can be manipulated. You can create an apnea or a cessation of breathing. Especially underwater, you don't want to be inhaling water. That would technically be drowning. So you create an temporary apnea by getting rid of the reason you want to breathe, which is the CO2. The brain has, we have a conscious brain that says we need to breathe, but we have control centers in our brain stem that are influenced by these conscious centers and also hypothalamus. And the brain stem has actually got the control centers that will tell you to breathe or tell you not to breathe. And they're usually responding to concentrations of CO2. For most normal people, normal conditions, it's CO2 that's driving breathing. You'd think it would be O2 concentrations, but O2 concentrations have to get very low before the brain is really concerned about O2 at all. And it will make more sense when we get into Sherwood why that is so. Most of the time we don't get our O2 levels to a critical level, even during heavy exercise. We still got some extra O2 that's available. And we want to get rid of the waste product, which is the CO2. We've got a respiratory rhythmicity center. That's going to set the pace of your breathing. And then we've got another center, it's called an apneustic center. Again, that A is implying cessation. So part of setting the pace is turn off the breathing. So you have a pace setter for inhalation, and then you've got a pace setter that's going to cause the exhalation. So you've got an inhale, and that fires to your external nerves, to your external intercostals and your diaphragm. And then after the volume changes, you've got an apneustic center that says you hit 500 mLs, turn off. And then those nerves are inhibited. And we've also got what's called a pneumotaxic center, which is adjusting this pace of breathing. So the number of breaths per minute may change. Maybe you're still getting 500 mLs in and out, but you're going from a normal 15 breaths per minute, maybe up to a higher or down to a lower breaths per minute. And that will be the job of the pneumotaxic center. In all of these, they're not working independently, they're working together to control your breathing. Pneumotaxic. They're not really all doing the same thing or opposite things. They're working together. And frankly, I don't think they've really figured out who is in charge up there. So we're going to talk a little bit about that. We've got these higher centers, your cerebral cortex, that is your conscious interpretation. I need more air, or I'm okay, I don't need to increase my breathing depth or rate. But you get nervous, your emotions start getting involved, and then that might change your respiratory rate. Whether you consciously are doing it, probably not, but subconsciously this emotional state you're in is altering the rate of your breathing and the depth of your breathing. Some people stop breathing when they get scared or nervous, and others hyperventilate and get shallower and more rapid breathing. And then hypothalamus, it's probably the most realistic, as long as it's healthy and functioning properly. It's probably the more reasonable controller of the brainstem. The higher center says, oh, I can hold my breath, I don't need to breathe. And kids who say, I want this, I want that, and if you don't buy it from me, I'm gonna hold my breath till I die. Yeah. Okay, honey. Sorry, see you go. You ain't getting it. Just sit down, put some pillows around them. Probably that's as far as you have to go, because they're going to say, what? You're going to let me die? That usually will stop them. But if they hold their breath and pass out, they're safe, because you have all the pillows around them. As soon as they pass out, they'll start breathing again, and then they'll wake up and go, oh my goodness, what just happened to me? And they probably won't throw that temper tantrum again. But if you give in to them, they gotcha. Okay, so higher centers, you can manipulate your breathing. Your emotions will manipulate your breathing, whether you want them to or not. Hypothalamus usually is just responding to information that's sent to it, and what the blood concentration of CO2 and O2 is. They will send action potentials to the pneumotaxic center. It's located in the pons. They can inhibit it, and then your respiratory pace decreases. They can excite it, and your respiratory rate will go up. So pointed arrow means excitation. The capacitor plate, double parallel lines, that means inhibition. So these higher centers can excite or inhibit your pneumotaxic center. Pneumotaxic center is going to be inhibitory of the apneustic center. So we've got double parallel lines there. So that means inhibition. So apneustic center is, what does it do? It excites this respiratory rhythmicity center in the medulla. So actually, kind of work backwards if you want to understand that. What does the respiratory rhythmicity center do? It's firing action potentials to your external intercostals and your diaphragm. So if you excite that respiratory rhythmicity center, then you're going to increase your breathing cycles. If this center, the apneustic center, is inhibited, then it's going to excite it's going to have a decreased excitation of your rhythmicity center, and your respiratory rate will go down. How do you decrease the excitation? By exciting your pneumotaxic center. Remember, it is inhibitory. So if you inhibit it, then there's less inhibition here, which means there's more excitation there, and in the respiratory rhythmicity center, and your respiratory rate will go up. If you excite your pneumotaxic center, then it's going to be a stronger inhibitor of the apneustic center. The apneustic center then is going to be less excitatory of your rhythmicity center, and your respiratory rate will go down. Sort of like the cerebellum situation in last semester, that we've got something that is inhibitory in its output. How do you get excitation? You inhibit the inhibitory center. and that releases the inhibition on whatever it's inhibiting. So it becomes more excited. We've also got feedback coming via your vagus nerve and vagus and glossopharyngeal nerve. Glossopharyngeal nerve and vagus, they're excitatory to the respiratory rhythmicity center. But the vagus nerve is also inhibitory to this apneustic center. So it's got a way of kind of grading the response or the effect that it has on the rhythmicity center. It excites it, but then it also can turn off the excitement from the apneustic center. So grading that output here back to your intercostals and diaphragm. Another excitatory influence on the rhythmicity center is your chemoreceptors. They're primarily the central chemoreceptors. They're responding to CO2 concentrations. They will not be looking at O2 concentration until O2 concentrations get to a critical level, and that's around 60 millimeters of mercury partial pressure. So very, very low. The CO2 and hydrogen ion, well, it's actually hydrogen ion that they're looking at, but it's monitoring CO2 indirectly. So CO2 is actually the drive for your breathing. For most of the time, the chemoreceptors are going to win out on the control. Okay, the debate goes on over here. You have a dorsal. This respiratory rhythmicity center is divided probably into more than just two centers. But we've got a dorsal and a ventral respiratory group, a DRG. Don't confuse that with the dorsal root ganglion. This is brainstem. This is a dorsal respiratory group. This is going to, and then a ventral respiratory group, a VRG. So dorsal respiratory group, where it's located near the tractus solitarius, which is part of our cranial nerve sensory pathways from last semester. It's going to drive the phrenic nerve. The phrenic nerve is, we learned last semester, the nerve that is going to the diaphragm. And when you excite the phrenic nerve, then the diaphragm goes up and down more rapidly. The dorsal respiratory group, now this is under debate, but nobody's disproved this. Nobody has offered anything concrete as an alternative, but it's the dorsal respiratory group is driving the ventral respiratory group. So it's exciting the ventral respiratory group. And then the ventral respiratory group is divided into a lower part and an upper part, which are called, the lower part is called the caudal division. The caudal division is inspiratory and then turns off for expiration, and the control for your intercostals. So inspiratory is going to be driving the external intercostals, and for expiration, it will be in control of the internal intercostals. The pathways in the ventral respiratory group, there are two essential pathways. We've got expiration that are crossing over to the opposite side. So they're going from the right brainstem to the left phrenic nerve, and the left brainstem to the right phrenic nerve. The inspiratory, you've got them duplicated. You got the right with one pathway going down on the right, being joined by one from the left. And then the one on the left has a pathway straight down and it's joined by one from the right side. My question to you, based on this, since expiration has one pathway and it's crossed, but inspiration has two pathways, one crossed and one uncrossed, which is more important, inspiration or expiration? Who says expiration? Who says inspiration? Inspiration wins. You're right. You can pretty much expire passively. If you can't use your abdominal muscles and your intrathoracic muscles, you can go up to a table and you can lean against it, or you can tell your friend to come and punch you in the tummy, and you can get a machine that will push the air out of you. But how are you going to get anybody to help you inspire? And you think about just passive breathing, you only work to inhale, and everything exhaling is passive. So inspiration is number one. You have to be able to do that. You can passively exhale with external help, or just turn off the muscles that help you to get that volume in. We're going to be relying on mechanoreceptors. These are our baroreceptors. They're measuring pressure in the airways, and they're going to be telling us about the change in our lung volume. We actually have stretch receptors in the lungs to help to prevent us from inhaling too much air. You don't think about the fact that it's possible to inhale too much air, but it is actually possible to do that. And so we have a stretch receptor that will automatically, like the fuse, you have a breaker switch. If there's a sudden surge of electricity through a certain pathway in your house, the breaker switch will shift off so that you don't fry the lines, the electrical lines in your house. We've got a breaker switch in our lungs that if for some reason, maybe it's a pathogen, toxin, or something, some chemical that you've ingested with a medication or self-medication, if for some reason you breathe in too much, you've got a breaker switch. The stretch receptor will shut off your inhalation and actually make you exhale more than you inhale, just to make sure you don't rupture these nice little alveoli that are down in the lungs. We've also got chemoreceptors in the lungs that will measure the concentration of carbon dioxide in the alveoli and oxygen in the alveoli. And actually, in case of carbon dioxide, they're responding to the pH change. So carbonic anhydrase has to be present to change the CO2 into the hydrogen ion. That can be measured by our chemoreceptors. We've also got, we told you about the breaker switch, this mechanoreceptor to prevent injury to your lung. We also have other sensors in there for pathogens that will initiate coughing or in upper respiratory tract sneezing that will be protective. So those are protective reflexes. As we age, everything gets less efficient, and that's including the lungs. The elastic tissue, like the elasticity of your skin, starts to decrease in the number of fibers, so your lungs don't stretch as easily, and you have to work harder to breathe that volume in. Also, the ribs start to get more connective tissue between them, and they get a little harder to move with your respiratory muscles. And if you get arthritis, then the joints that are between your vertebrae and your ribs and your sternum and your ribs, those can get affected by arthritis, and it becomes painful to breathe. Emphysema is a condition caused by a lot of different things, but mostly it's smoking that causes emphysema. When you get emphysema, you actually lose the membranes of your alveoli. You start to get larger alveoli, which means you're losing the elastic connective tissue that was there. The lung becomes less elastic. It also, because of the decrease in the wall space, you get less oxygen and CO2 exchange, so less surface air for exchange. And also the lack of this elastic connective tissue affects your bronchioles. You wouldn't think about that as being a possibility, but you got bronchioles, those openings. They're not that big, of course, but these are smooth muscle that is keeping those airways open. They're physically attached to all the little alveoli out here, which extend out to the surface, to your pleura, and the pleura is attached to your chest wall via the pleural cavity. And also to the membrane this side. So all these alveoli, they're tethered to each other. They're tethered to the bronchioles, and the bronchioles are tethered to the chest wall. When this tethering, when this ability to attach the bronchioles to other structures decreases because you start losing alveoli and you get less elasticity going on, then the airways tend to have a smaller diameter and you get less oxygen in and out. So in emphysema, when you lose that elasticity and then lose the tethering effect on your bronchioles, if you exhale forcefully, your bronchioles will collapse before any air gets out of your remaining alveoli. And you trap a lot of air in your lungs. Your residual volume goes up. Doesn't seem like it would be too big a deal, but you have some dire consequences. Your quality of life is going to change. You're not going to be able to exercise as much. That kind of goes with aging, but this is extreme. It's beyond what normal aging would produce. Also, when you have a lot of air that's trapped down in there, get one little bacteria down in there and trap it down in there with, if it's a gram-negative bacteria, or if it's anaerobic bacteria and you don't get a lot of oxygen down into those alveoli because you don't have the same flushing of new air in and out, they just thrive down there, and now you've got a lot of infection going on in the lungs. So it can have dire consequences. As we age, somebody has figured out that we're losing about 1 square foot of respiratory exchange surface for each year after age 30. So you guys aren't too far from age 30. You're on the downhill side for your respiratory tract from then on. Doesn't really... Is that me buzzing? Is that you? Anyway, I have mine on vibrate. So you don't have to be limited there are, you exercise, you can maximize the volume that you do have, and not, it doesn't have to be a really precipitous drop, but that's a lot of